Hello students, today's class is on lymphatic system. In this class, we'll study mainly the anatomy of the lymphoid tissue and some of the cells of the lymphatic system. The competencies are, we'll study the components of the lymphatic system, we'll study the circulation of lymph in the body, then we'll study the humoral and cell-mediated immune responses, the cells that are forming the structural and functional constituents of the lymphatic system, and various lymphatic organs of the body and their major histological features. The lymphatic system, it starts as blind capillaries in between the cells. When a bacteria, virus or antigen enters into the gut, they are absorbed through the macromolecules, actually the molecules which are not able to pass into through the uh, blood capillaries, they are carried by lymph capillaries. The lymph capillaries actually starts as blind spaces in between the blood vessels. So the bacteria, virus or the antigen, they pass between the cells and accumulate in the intercellular spaces. So the lymph capillary starts there only in between the cells, and they join to form the bigger lymph vessels. The bigger lymph vessels passes mainly along with the blood vessels, and they carry lymphatics from the body and they enter into the lymph nodes. Lymph nodes may be isolated lymph node or they are in the groups. So they enter into the lymph node and they pass through the lymph node. The efferent lymph vessel comes out from the hilum of the lymph node and it carries immunocompetent cells or lymphatic cells along with the lymph, and they join and form bigger lymph vessels. As you have seen, the thoracic duct, the thoracic duct drains into the larger venous system. So ultimately, the lymph joins the venous system. The lymphoid organs and the lymphatic vessels in the body, there are central lymphoid organs. There is the thymus, which already you have dissected. Then the bone marrow, where all the lymph cells are produced in the bone marrow. The spleen is one of the peripheral lymph organ. The inguinal lymph nodes are a group of peripheral lymph organs. Then you already have dissected the axilla and you have seen a lot of lymph nodes in the axillary region. Those are all peripheral lymph organs. In the inguinal region also there are superficial and deep inguinal lymph nodes along the femoral artery, and there are MALT or mucosa associated lymph nodules which is present in the submucosa of the gut, bronchi, urinary system and the female reproductive system. Origin and maturation of lymphocytes. Lymphocytes undergo antigen-independent differentiation in the primary lymphatic organs. In humans and other mammals, the bone marrow and the gult or mold, together the Barca equivalent organ, and the thymus has been identified as primary or central lymphatic organ. Lymphocytes differentiate into immunocompetent cells in these organs. Initially, lymphocytes are genetically programmed to recognize single antigen out of virtually an infinite number of possible antigens. A process called antigen independent proliferation and differentiation. These immunocompetent cells then enter the blood or lymph and are transported throughout the body where they are dispersed in the connective tissues. The immunocompetent lymphocytes, together with the plasma cells from B lymphocytes and with macrophages, organize around the reticular cells and reticular fibers to form the effector lymphatic tissue and organs, that is lymphatic nodules, lymph nodes, tonsils and spleen. Within this secondary or peripheral lymphatic organ, T and B lymphocytes undergo antigen-dependent activation into effector lymphocytes and memory cells. Therefore, basically the lymphoid system has lymphoid organs that may be central, that is thymus and the bone marrow, and peripheral, that is the lymph nodes, spleen, submucosal lymphoid tissue in gult or mold, and the cells are lymphocytes, B cells, which is responsible for the humoral immunity. They produce antibodies and react with antigens. The T cells, they are responsible for the cell-mediated immunity and they express CD molecules or cluster of differentiation molecules on their surfaces. We'll come across later what is CD. There are natural killer cells. Along with the B and T cells, there are natural killer cells. We'll come across later the details about the NK cells. Basic type of immune reaction. In cellular immunity, the cells react against and kill the microorganisms, foreign cells from tumors and transplant, and virus infected cells. This category of immunity is mediated by T lymphocytes or T cells. Humoral immunity. Humoral immunity is related to the presence of circulating glycoproteins called antibodies that inactivate or destroy the foreign substances. The antibodies are produced by plasma cells derived from B lymphocytes or the B cells. Innate immunity. The immune reaction can also be innate or adaptive. It is also called acquired immunity. The innate reaction is simplest response and the response is very fast, non-specific, and does not depend on previous contact with pathogens. The main cells responsible for the innate reaction are phagocytes, that is macrophages, neutrophils, and natural killer cells. Even though they are not part of the immune system, the barriers formed by epithelial cells of the internal linings and the skin protect the organism against the penetration of foreign molecules and microorganisms. The adaptive reaction is also a refinement of the more primitive reaction. The adaptive reaction is more efficient, highly specific, and has memory. A second attack of the same pathogen is dealt by faster and more efficient cells, because it is not only it involves not only the same cells as the innate reaction, but also the formation of antibodies, and adaptive reaction starts more slowly. There are certain terms you have to remember. What is an immunogen? These are foreign substances encountered by immune system and that acts as immunogen. That is a substance that elicits a response from the host. Then comes antigen. More specifically, an antigen can react with antibody even if it is not capable of eliciting an immune response. Because most of the immunogens are also antigens. Classically, the immunogens are also called antigens. The specificity of the humoral immune response by the B cells is determined by small molecule domains or antigenic determinants or epitopes of the antigen, whereas specificity of the cellular immunity or immune reactions by the T cells is determined by small peptides associated with major histocompatibility complex, which we will discuss later, which is present on the membrane of the antigen presenting cells. An antigen has many epitopes and will react widely or has wide spectrum reaction with the humoral and cellular responses. Antibodies. The antibodies are also called immunoglobulins, are circulating plasma glycoproteins that interact specifically with the antigenic determinants that are elicited in their formation. Antibodies are secreted by plasma cells that arise by proliferation and differentiation of the B lymphocytes, and many of the antibodies you know, the immunoglobulins IgG, A, M, D, E, etc. Cells of the lymphatic system. Already we know there are three types of cells or three types of lymphocytes. These are the B lymphocytes, T lymphocytes, and NK cells. And there are supporting cells in the lymphatic system. These are reticular cells which synthesize reticular fibers to form the framework of the lymphoid organ, where lies the monocytes, macrophages, neutrophils, eosinophils, basophils, dendritic cells, follicular cells, epithelio-reticular cells, Langerhans cells. The epithelio-reticular cells are special types of cells present in the thymus which forms the framework of the thymus. The Langerhans cells are present in the epidermis of the skin. Dendritic cells or the follicular cells has long processes like dendrites of a neuron, and they are present in the spatial regions of the lymph nodes. There is a term called CD or cluster of differentiation. This is an international nomenclature which is unique cell surface molecules expressed during the differentiation or activation. The nomenclature is like CD1, CD2, CD3, CD4 onwards. T and B cells are morphologically similar. They are different in their site of maturation and receptors. T lymphocytes. There are three types of T lymphocytes. T cytotoxic lymphocytes. They carry a receptor or T cell receptor on the surface. They express CD8 markers. They are specialized to recognize antigens associated with MHC or major histocompetitive complex 1 on the surface of other cells. They produce perforin and other proteins that kill the foreign cells, virus infected cells, and some tumor cells. They induce apoptosis. T helper lymphocyte, it also carries T cell receptors. It expresses CD4 markers. It secretes cytokines and modulates other T and B cells, stimulating their activities. T memory lymphocytes, it also carries T cell receptors on the surface. It is primed to respond more rapidly and to a greater extent upon subsequent exposure to the same antigen. It stimulates production of cytotoxic T cells. The gamma or delta T lymphocytes, they do not express CD4 or CD8 markers. They develop and mature in the thymus. They migrate to the skin or mucosa to encounter antigens. The B lymphocytes, NK cells and APCs. The B lymphocytes carries membrane receptors, that is IgM. When they are activated by specific antigens, they proliferate by mitosis. They differentiate into plasma cells that secrete large amount of antibodies. T helper cells produce interleukin 2, induce proliferation of B lymphocytes to plasma cells. B memory cells. These B memory lymphocytes are activated B cell that is primed to respond more rapidly and to a greater extent on subsequent exposure to the same antigen. NK lymphocytes or NK cells, they lack T and B cell receptors and it attacks the virus infected cells and cancer cells without previous stimulation. Antigen presenting cells or APCs, they are present in the skin. These include dendritic cells, macrophages, Langerhans cells, B lymphocytes. They phagocytose and process antigen and present the antigen to T lymphocytes to induce their activation. Percentage of lymphocytes in various lymphatic or lymphoid organs. The thymus contains totally 100% T lymphocytes and B lymphocytes are absent in thymus. Bone marrow contains 10% T lymphocytes, 90% B lymphocytes. The spleen contains 45% T lymphocytes and 55% B lymphocytes. Lymph nodes contain 60% T lymphocytes and 40% B lymphocytes. In the blood, there are 80% T lymphocytes and 20% B lymphocytes. Let us know certain characteristics of dendritic cells, major histocompatibility complex or MHC. The dendritic cells, the origin in the bone marrow and the lodge in the lymph organs or in the skin. In the T cell region of the lymph nodes, they are called dendritic cells or follicular dendritic cells. These dendrites capture and process antigen and initiate immune reaction. The major histocompatibility complex or MHC, they are the surface molecules present in all cells. In humans, it was first discovered in leukocytes. That's why it is called human leukocytic antigen or HLA. They are unique for each individual and it is basis for tissue transplantation. MHC1 is present in all cells, whereas MHC2 is present in APCs or antigen presenting cells. Lymphocyte development and differentiation. All the lymphocytes take origin or they're born in the bone marrow. The primary or central lymphatic organs are the bone marrow, GALT, gut-associated lymphoid tissue, thymus. Lymphocytes become immunocompetent in these areas. After that, they enter the blood and disperse in the connective tissue. Secondary lymphatic organs, lymphocytes after taking origin, they circulate in the blood and they reach the lymphatic nodules, lymph nodes, tonsils, spleen. T and B lymphocytes undergo antigen-dependent activation into effector lymphocytes and memory cells. The diseases of the lymphatic system. Allergic reactions. Most of you have suffered from some or other form of allergic reactions. These reactions are intense abnormal reactions in attempt to neutralize the effects of an antigen, and hypersensitive reactions are severe type of allergic reactions. There is immune deficiency or there is depression of the reaction against the antigen due to defect in the T and B lymphocytes or complement system or phagocytosis. And there is autoimmune disease. Tissue destroys by their own T cell against own body organs or against itself. So there are three types of diseases in the immune system. Allergic reaction, any allergen which is foreign to the body, if it enters, there will be a reaction, and a severe reaction to any substance is hypersensitivity reactions. It may happen to any insect bite or any drug. Immune deficiency, there is deficiency of the, uh, the lymphocytes. And autoimmune disease, the lymph node acts against the body, the own body, that is autoimmune disease. After going through the cells and organs of the lymphatic system, let us see the histological features and gross features of the different lymphoid organs. First, we will discuss about the thymus. Thymus is bilobed gland which is situated in the thorax, just behind the sternum, in front of the arch of aorta and its branches. The lobes may go higher up to the lower pole of the thyroid gland, or in the lower part it may cover the anterior surface of the heart. Developmentally, thymus develops from two sources, the endoderm and mesenchyme. Endoderm form the framework of the thymus, and the mesenchyme forms the lymphocytes of the thymus. The thymus is covered by thick capsule which sends trabecularly inside the gland, dividing the gland into smaller lobules. This trabecule carries the blood vessels of the supplying the thymus. If you examine the lobule, there is a peripheral part which is darker in color and a central part which is lighter in color. The peripheral part is called cortex. It contains small T lymphocytes, and the central part is called medulla. It contains large T lymphocytes and Hessel's corpus. The medulla of the thymus is lighter in color and it contains certain eosinophilic areas. Those are the epithelio-reticular cells which takes lighter color, and thymic corpuscles. Thymic corpuscle contains concentric layers of epithelial cells or epitheloreticular cells. The central part is degenerated or sometimes calcified. This is a characteristic feature of the thymus. The function of these SS corpuscles are not known, but they are the degenerated epitheloreticular cells which forms the framework of the thymus, and within these frameworks lies the T lymphocytes. The blood supply of the thymus is of special importance. The blood vessels from the cortex, they pass into the trabeculae. From the trabeculae, the blood vessels pass to the junction between the cortex and medulla as arteriole. This arteriole gives capillaries or arch capillaries which enter into the cortex. The medulla is supplied by the capillaries, and from there the venule starts. Venule enters into the trabeculae to form larger veins. Blood-thymus barrier requires special mention as it doesn't allow the antigens to reach the thymus, or it protects the T cells from the exposure of various antigens. The blood-thymus barrier is composed of capillaries which is present in the cortex. Just like any other capillaries, it has endothelial lining which lies on a basement membrane or basal lamina. The basal lamina is very thick in the thymus or in the capillaries of the thymus. It has also pericytes. The second layer of the blood-thymus barrier is formed by the perivascular connective tissue, which is synthesized or formed by the macrophages, and it contains some collagen fibers and reticular fibers. The third layer is formed by the epithelio-reticular cells. It is called epithelio-reticular layer, and outside that there is T lymphocyte. The blood-thymus barrier is absent in the medulla, and it protects the T lymphocyte from antigenic exposure. The T lymphocytes mature in the thymus. 95% of the T lymphocytes, they die by apoptosis. Only 5% differentiate and are released to the circulation. The thymus is well developed in a newborn. The weight of the thymus in newborn is 10 gram, whereas during puberty we may reach up to 20 gram. After puberty, the thymus starts involution, and in adult a small amount of thymic tissue is present. The rest of the thymus is composed of adipocytes. Functions of thymus: development, differentiation, and maturation of immunocompetent T cells from the precursors derived from the bone marrow, or the stem cells for the T cells are developed in the bone marrow, and they are differentiated and become immunocompetent in the thymus. There are T cell dependent zones in the lymph node. There is a paracortical region of the lymph node, sphenic periarterial sheath, and also pairs patches. The second function is proliferation of clones of mature or naive T cells for circulation in the peripheral tissues. Development of immunological self-tolerance also, and it also secretes certain hormones and certain other factors for the growth and development of the thymus. These are thymolin, thymopoietin, thymosin alpha, etc. After examining the thymus, let us examine the structure of the lymph nodes. The lymph nodes are bean-shaped bodies which are present throughout the body. Nearly 600 to 650 lymph nodes are present in the whole body. They are in groups or in isolated uh, positions. Groups of lymph nodes you have already seen in the axilla, groin. They are also present in the abdomen and thorax. In the abdomen, they lie around the abdominal aorta. In the thorax, you have seen in the hilum of the lung, in the mediastinum, in the neck also, there are deep and superficial groups of lymph nodes. The lymph nodes are bean-shaped with a hilum through which the blood vessels enter and the efferent lymph vessels come out. The lymph nodes are covered by a thick capsule which sends branches inside the substance of the lymph node. Those are called the trabeculae, through which the blood vessels enters inside the lymph node, the smaller blood vessels. The afferent lymph vessels, they pierce the capsule and they drain the lymph in the subcapsular sinus. These trabeculae divide the lymph node into different compartments. The outer cortex contains the lymphatic nodules, those are called cortical nodules, and the inner part or inner cortex. The innermost part is called medulla. So, structurally, the lymph node has three parts: the outer cortex, the inner cortex, and the medulla. The inner cortex is present between the outer cortex and the medulla. The blood vessel enters through the hilum and it divides and supplies the nodules. The venules form and they pass through the trabeculae to form the bigger veins which come out through the hilum. The afferent lymphatics drains into the substance of the lymph node, and the efferent lymph node vessels, they start from the bigger spaces inside the medulla or the medullary sinuses. They join to form bigger lymph vessels. They can come out through the hilum. This is the artery, this is the efferent lymph node, and this is the vein. This is a real section of a lymph node stained with hematoxylin and leucine, showing the peripheral part or the cortex, which is deeper in color, containing the lymph nodules, and the inner part, which is lighter in color, is the medulla. And the junction between the cortex and medulla is called the deep cortex. There is a rich capillary plexus, which are the branches of the artery which enters through the hilum and divides into arteriole and capillaries, supplying the lymphatic nodules. This is an enlarged picture of the lymph node showing a thick cortex covered by capsule and the cortex containing the germinal centers or lymphatic nodules. The center of the germinal center is lighter in color because the center contains large lymphocytes, and the peripheral part is darker in color. The B lymphocytes are present in the central part of a lymphatic nodule. The capsule sends some connective tissue fibers. Those are called the trabeculae, which is dividing the lymphatics into outer part and the inner part. In the medulla, there are medullary cords which contains especially the lymphocytes, macrophages, reticular fibers, and the medulla also contains B lymphocytes. The T lymphocytes are present in the inner cortex. So, in short, the B lymphocytes are present in the lymphatic nodule. T lymphocytes are present in the inner cortex, and the medullary cords also contains B lymphocytes. And there are spaces in between the medullary cords. Those are called the medullary sinuses, which contains lymph, which is carried from the subcapsular sinus to the medullary region. This is thick capsule, and just below the capsule is the subcapsular sinus containing the lymph and the cells of the lymphatic series, especially the lymphocytes, macrophages, the reticular cells, and this is the outer part of the cortex, which is one of the part of the lymphatic nodule. This is showing the part of the activated lymph node. In inactivated lymph node, the nodules are totally darker in color. In activated lymph nodes, the nodules are the central part of the nodules are lighter in color because of the large lymphocytes, macrophages, and dividing cells are present in the center. This is also showing the medulla of the lymph node, showing the medullary cords which contains small lymphocytes, the reticular cells, macrophages, and in between there are spaces. Those are called the medullary sinus. These are medullary cords, and in between. This is a higher picture of the medulla of a lymph node showing the different cells of the medulla. It contains macrophages, the larger cells, smaller lymphocytes, and the reticular cells with long processes. Lymph circulation in a lymph node. The afferent vessels or afferent lymph vessels pierces the cortex, and the lymph vessels drain into the subcapsular sinus carrying the lymphocytes. The lymph and the lymphocytes from the subcapsular sinus, they pass to the trabecular sinus by the side of the trabeculae. The lymph from the subcapsular sinus also enters into the germinal center where it is differentiated and become immunocompetent. They leave the germinal sinus and they again come to the subcapsular sinus in the deep part of the cortex, where you see the high endothelial venules. The deepest part of the lymph node is the medulla, which contains the medullary sinus. The subcapsular sinus drains into the medullary sinus in between the medullary cord. This medullary sinus join to form the efferent lymphatics or efferent lymph vessels. The lymphocytes also come along with the arteries, and it passes to the germinal centers where the capillaries form. The venules are also formed from there. The venules pass to the deeper part of the cortex, and the special type of venules are there. These are the high endothelial venules through which the lymphatics or the lymph passes in between the endothelial cells to the substance of the lymphatics or substance of the lymph nodes. This picture shows the detailed circulation of the lymph through an artery. It enters into the hilum, then it passes into the germinal center, then the high endothelial venules are there in the deep part of the cortex. Here it comes out. It comes out of the high endothelial venules into the substance of the lymph node. Then it enters into the subcapsular sinus. Then they collect to form the efferent lymph vessels. Then bigger lymph vessels drain into the veins. The high endothelial lymph vessels are present in the lymph node and the peripheral lymphoid tissues, but they are absent in the spleen. This is showing the high endothelial venules and the gap between the columnar or cuboidal epithelial lining of the venules. Through these gaps, the lymphocyte passed from the lumen to the substance of the lymph node. The spleen. The spleen is hemolymph organ. It is present in the abdomen. It is covered by a capsule and it has a hilum through which the blood vessel enters and the veins come out. The capsule of the spleen is very thick and it sends branches inside the substance of the spleen. Those are called the trabeculae, which divides the spleen into different parts, and through this trabeculae blood vessels pass into the substance of the spleen. The spleen has collections of lymph nodules, and in between the collections there are red areas. Actually, in fresh spleen, in gross structures, if somebody cuts the spleen, one can see certain white areas and certain red areas. That's why name has been given white pulp and red pulp. The white pulp contains lymphatic nodules and the red pulp contains the, the splenic cords and RBCs. This is the capsule trabeculae, the white pulp and the red. This is a detailed structure of the white pulp and red pulp. The white pulp is just like a lymphatic nodule of a lymph or lymph node, which has a central lighted area and peripheral darker area. and in the eccentric position there is a blood vessel. This is called the central artery of the spleen, and very surrounding the white pulp is the red pulp, which contains the splenic cords and the RBCs. This is the central artery. The splenic circulation. The splenic artery divides and subdivides, enters into the trabecule to form the trabecular artery. The trabecular artery gives branches to white pulp, that is called the central artery. The central artery, when it enters into the white pulp, it gets a sheath of T cells, and these are called periarteriolar lymphatic sheath, PALS. Then the trabecular artery leaves the white pulp and it gives peninsular arterioles. These peninsular arterioles drain into the splenic sinus, or the peninsular arterioles are also covered by the sheath of lymphoid tissue. There are two theories regarding the splenic circulation. The closed circulation. The peninsular arteries directly enters into the splenic sinuses, and from the splenic sinus or sinusoids, the venules start and the bigger veins are formed. The bigger veins join the trabeculae to form the bigger splenic veins, which comes out through the hilum. The open theory is that the peninsular arteries opens into the red pulp, and the contents of the arteries enters into the spaces in the red pulp. Then they enters into the splenic sinusoids. The sinusoids uh, drain into the venules, which forms bigger veins. This is showing in details of the splenic circulation. The trabecular artery, when it enters into the white pulp, it gets a sheath of T cells. This is called periarteriolar lymphatic sheath. Then, when it comes out from the lymphatic nodule, they are formed, so they are called the peninsular arteries. Peninsular arteries also are ensheathed by lymphocytes. In closed circulation, they directly enters into the splenic sinus, and according to the open circulation, they enters the terminal arteriole of the splenic capillaries, ends into the red part. This is closed circulation, this is open circulation. This is also diagrammatic representation of closed circulation. These are the splenic cords. The splenic cords contains lymphocytes, connective tissue, reticular fibers, and in between there are spaces, and in those spaces only the blood enters the spaces between the splenic cords, and from those spaces again blood enters through those gaps into the sinusoids. So this is the open circulation. In closed circulation, from the peninsular arteries directly arterioles and venules are formed, and in between the splenic sinusoids are there, so the arterioles directly drain into the splenic sinusoids. This is closed circulation, open circulation. The functions of the spleen. The major function of the spleen, just like any other lymphatic or lymphoid organs is uh, production of immunocompetent cells, production of lymphocytes, and there are lots of macrophages are there, and those macrophages, they engulf the worn out all the old RBCs. The RBCs, this is the graveyard of RBCs. The dead RBCs are broken down by the macrophages, and the hemoglobin is uh, broken here to form the heme and the other proteins also. So main function is production of lymphocytes, destruction of erythrocytes, and defense against invaders. This is showing the RBC inside the macrophage. Certain question arises: what is myeloid metaplasia and what are the effects of splenectomy? The person can survive without spleen also, although this is a graveyard of RBCs. When the spleen is removed, the RBCs are destroyed in the bone marrow and liver. This is a detailed picture of the spleen showing the white pulp or the capsule first, then the white pulp showing the lymphatic nodules, and typical lymphatic nodules, the inner part is lighter, outer part is darker. Inside there is trabeculae, and there are blood vessels, the trabecular veins are there. So these darker parts are the white pulp and the lighter part are the red pulp. This picture shows the branches of the splenic artery, how it is going through the splenic nodule. Then the open circulation and closed circulations are there. The splenic sinusoids are there. From sinusoids, the venules are formed, the bigger veins are formed. This is the splenic nodule. This is a trabecular vein. This is red pulp. This is the central artery. This is a higher picture of the spleen showing the splenic nodules or the germinal centers. This is a typical white pulp. The lymphatic nodule, the central part is lighter because of the larger cells. The peripheral part is darker because of the smaller cells, and the artery or the central artery is not present in the center but slightly outside the center. The mucosa associated lymphatic nodule or the MALT. Our oral cavity and the respiratory channels or respiratory tract is always exposed to external world. So there are collections of lymphocytes around the entry of the pharynx and also in the mucosa of the whole of the GI tract and the respiratory tract. There are tonsils at the back of the mouth. Those are called Waldeyer's ring of tonsil. The ring of tonsil is components are the tonsil itself. There is lingual tonsil on the tongue. There is pharyngeal tonsil on the back of the pharynx. The structure of the tonsil is that it contains lymphatic nodules, and according to the situation, the epithelium is either stratified squamous or columnar, or in case of the bronchial tree, it is pseudostratified ciliated columnar epithelium. Here it is showing a part of the bronchial epithelium deeper inside the lung, showing the lung tissue and the bronchiole The epithelium of the bronchial is here, and deep to the epithelium there is a collection of lymphoid tissue. This is called mucosa associated lymphoid tissue or MALT. Just like lymph node or other places, it has lighter part of the nodule and peripheral part is the darker part. This is the lymphatic nodule in the small intestine. There is collection of lymphatic nodule in the small intestine called Pear's patch. So this is one of the nodule inside which you can see the central part is lighter and the peripheral part is darker. These are the intestinal villi. This is diffuse lymphatic tissue in the lamina propria. The lamina propria is just beneath the mucosa. There are lots of free lymphoid cells, especially it contains the plasma cells, macrophages, and small lymphocytes. This is one of the gland of the intestine. This is another one. The lamina propria is the place where there are lots of lymphoid cells. This is palatine tonsil by the side of the oral cavity at the beginning of the pharynx. The palatine tonsil is situated. Again, you can see lot of lymphoid nodules are there. The central part is lighter and the peripheral part is darker in color. So one of the characteristics of the palatine tonsil is that there are deep grooves, and those grooves are called tonsillar crypts. As it is a part of the oral cavity, it is lined by stratified squamous epithelium. Please remember that um, the tonsil doesn't have a capsule on towards the, it doesn't have a capsule towards the oral cavity. Part of the capsule is there away from the oral cavity, and the tonsillar epithelium is stratified squamous epithelium. So there are deep grooves which are called tonsillar crypts. The tonsillar crypts are also lined by stratified squamous epithelium. So wherever you see a lymphatic nodule and stratified squamous epithelium, it is palatine tonsil. This is the aggregated lymphatic nodule in the wall of the ileum, and those are called the Pear's patches. Again, you can see lymphatic nodule containing the lighter area and peripheral part is the darker area. This is a part of the lymphatic nodule in the small intestine. It's a part of the Pear's patch. So, special type of cells called M cells. The M cells are, are antigen presenting cells in the small intestine. Here, the part of the small intestine which contains macrophage, and inside the macrophage one can see a large number of bacteria are there. This is actually in the lamina propria of the small intestine where the collection of lymphocytes are there. Those are called Pear's patches. This is mucosal immunity in the small intestine it is showing. So when a antigen is or antigen comes in contact with the enterocytes, then what happens? The mm, macrophages engulf the antigen, and there are lots of T and B lymphocytes and dendritic cells. These become activated and they go inside the circulation where the other lymphocytes are activated. Then B lymphocyte here uh, starts production of antibodies, and the macrophages engulf then activate the T lymphocytes also. So the local immunity is there, which which uh, uh, works against the antigens, and they don't allow the bacteria or virus to pass into the circulation.